was hoping to share just one example with you today around the PD-1 pathway. Obviously, many of you are aware this is a pathway that has transformed oncologic therapies in the development of immunotherapies now that reawaken the immune system to target cancer cells. It's been very challenging, however, to evaluate which individuals are going to benefit from these PD-1 therapies. And we know clinically there's massive heterogeneity, once again, in responders versus non-responders. This is an area we've thought about for quite some time. And if we look at those genetic common polymorphisms, again, that occur in the PD-1 pathway, we can ask across the 50 to 100,000 circulating factors that we've assayed in our underlying database, which ones associate with polymorphic variants in the PD-1 pathway. And when we look, we find there's 4 to 5 individual markers, again, that are shown here on this bottom chart that are strongly and statistically associated with changes in the PD-1 pathway. Now we can take one of these markers, isolate it out, once again, solve its underlying structure, develop targeted assays. And when we examine this in individuals undergoing immunotherapy, what we find on here on the left is relative to pretreatment. This is a marker that's highly dynamic in increases in individuals undergoing a PD-1 or PD-1L-like therapies as part of their immunotherapy regimen. Now, even in those individuals that have an elevation in this marker, what you'll see on the graph on the left is there's quite a bit of spread in the post-treatment measurement, and that there's some individuals that have very high levels of this, indicative of greater target engagement, and individuals that have a more modest increase in this pharmacodynamic or target engagement biomarker, indicative of less target engagement. Now, if we ask those individuals that have the most robust biomarker response, how does this map onto actual response to the underlying therapy? What you find is quite startling. And that is those individuals that have greater evidence of target engagement, again, as assayed by this single biomarker, have almost a 50% increase in response to the underlying PD-1 therapy, as shown here on the right. Again, this is a marker that we can begin to interrogate in our human biological database, beginning to ask many factors and understand the biology of this. We've already begun to understand its response in immunotherapy, as well as changes across various demographics, how this maps onto other autoimmune and immune-related function, including over time, understanding how these change over the course of a 24-hour period in relation to cytokines and other inflammatory states, as well as to understand how these markers may be altered in both acute infection, vaccine response, and in relation to various immune cellularity as a means providing an understanding of how these markers may be reading out underlying immunotherapy response.